Good afternoon. So the next enzyme we are going to study, we have finished with restriction endonucleases, methylases, kinases, and polymerases. The next enzyme is phosphatases. So there is very much less difference between kinases and phosphatases. These phosphatases cleave the phosphate group from substrate by using a water molecule. Kinases, they donate the phosphate group from one substrate to another substrate. So phosphatases, they catalyze the breakdown of phosphate group from a substrate and they make use of a water molecule for that purpose. It is also known as hydrolytic cleavage. This reaction is not reversible. So exactly the opposite activity of kinase or and phosphorylase. So they are totally having opposite activity. Which add phosphate group to the substrate, and phosphatases, they remove the or they break down the phosphate group from the substrate. So there are two types of phosphatases based on their activities: acid phosphatases and alkaline phosphatases. You can hear, no? Can you hear, Shruti? बाकीचे जॉईन नाही होत का? बाकीच्यांना लेक्चरची आवश्यकता नाहीये वाटतं. 1 2 3 4 5. Okay. So acid and alkaline phosphatases. The most common form of phosphatases are alkaline phosphatases. So the special class of phosphatase is there, which is helpful in the removal of phosphate group from proteins. The substrate here will be phosphate, and they are called phosphoprotein phosphatases. So acid phosphatases, alkaline phosphatases, and phosphoprotein phosphatases. Okay, so you see the substrate here, water molecule is lost, water molecule is added. With the help of this water molecule, the substrate gets the OH group from this water molecule and phosphate is released out. This remaining H comes and joins here. So 3 O minus, O minus, O minus, and ithe OH. So this is actually the mode of action of phosphatases. These phosphatases, they remove the phosphate from the substrate with the help of a water molecule. Acid phosphatases. So the optimal activity definitely will be between 3 and 6 because they are acid phosphatases. Example is lysosomal enzyme. It catalyzes the hydrolysis of organic phosphates and it liberates one or more phosphate groups. Usually, these type of enzymes, they are found in prostatic epithelial cells in the prostate glands, erythrocytes, then tissues of prostates, spleen, and kidney. There usually these type of removal of one or more phosphate groups takes place by water molecules. So they hydrolyze organic phosphates and they liberate one or more phosphate groups. This activity is more common in the prostate glands. Then alkaline phosphatases, they are homodimeric enzymes and they catalyze the reactions like hydrolysis, transphosphorylation of phosphate monoester. They show their optimum activity at pH 10, obviously because they are alkaline in nature. Alkaline phosphatase was the first zinc enzyme discovered and it has three closed space metal ions: zinc ions, magnesium ions, two zinc ions, and one magnesium ions. So these were the first zinc ions, zinc enzymes, and they consist of three closely placed metal ions, two zinc ions, and one magnesium ions. So these are the bridges which are held together by ASP51. That is the amino acid aspartic acid at 51 position. The mechanism of action is based on the presence of covalent serine phosphate intermediate and which is formed to produce the inorganic phosphate and alcohol. So हा जो मेन रिमूव्हल ऑफ फॉस्फेट ग्रुप आहे तर तो कुठे एक्झॅक्टली होत असतो? सो ॲट द covalent serine phosphate intermediate is first formed. Serine will be the amino acid which will be present in alkaline phosphatase. आणि तिथे तो phosphate intermediate attach होतो. A temporary serine phosphate intermediate will be formed and it will liberate the phosphate and alcohol. In human body, alkaline phosphatase is present in 4 isoforms, in which 3 are tissue-specific isoforms. So they are present in special tissues. Isoforms means what? Structurally similar, functionally they may be functionally similar, but they may be some modification, isomeric forms. So here, depending on their location, they have their function, and so they are known as tissue-specific isoforms. That is in the placental barriers, germ cells, intestinal cells, and one is a non-tissue specific isoform. So 3 टिश्यू स्पेसिफिक आहेत कारण ते त्या टिश्यूज मध्येच दिसतात. एक प्लासेंटा मध्ये, एक जर्म सेल मध्ये आणि एक इंटेस्टिनल सेल्स मध्ये आणि एक जनरल आहे. सो इट इज नोन एज नॉन टिश्यू स्पेसिफिक. The genes that code for tissue specific isoforms are present on the chromosomes on chromosome 2. So they are P37, Q37, while the genes for non-tissue specific are present on chromosome 1. Chromosome 1 वरती genes असतात ते non-tissue specific phosphatase तयार करतात, while chromosome 2 वरती tissue specific isomeric form of phosphatase तयार करण्याची क्षमता असते. During post-translational modification, alkaline phosphatase is modified by N-glycosylation. So आपण ट्रान्सलेशन मध्ये बघितलं की खूप सारे पोस्ट ट्रान्सलेशनल मॉडिफिकेशन, ग्लायकोसिलेशन, नायट्रोसिलेशन, ऍसिटिलेशन वगैरे असतं. त्यात हे ग्लायकोसिल ग्रुप अटॅच होण्याचं काम आहे. And that is again a post-translational modification. So alkaline phosphatase enzyme will be modified at that time after its synthesis from chromosome 1 or chromosome 2, and the modification is N-glycosylation. It undergoes modification through uptake of 2 zinc ions and 1 magnesium ion. या ग्लायकोसिलेशन प्रोसेसच्या वेळी 2 झिंक आयन्स आणि 1 मॅग्नेशियम आयन्स त्याच्यात अटॅच होतात and that forms the active site of the enzyme alkaline phosphatase. These can be isolated from various microorganisms. Tissue specific असतील तर त्या tissues मध्ये हे enzymes आपण isolate करू शकतो. Connective tissues of invertebrates and vertebrates and in the human body. Germ cells आहेत, placental barrier आहे तर तिथे जर हे present आहे तर तिथून आपण त्यांना isolate करू शकतो. Action of AP, alkaline phosphatase. So there are several alkaline phosphatases which are used in gene manipulation in rDNA technology. One is bacterial alkaline phosphatase. So it is a phosphomonoester which hydrolyzes 3' 5' phosphate from nucleic acid. So it removes that 3' 5' phosphate from DNA. It removes the phosphate group before end labeling, and it can remove phosphate from vector before the insertion and ligation. ज्यावेळी लायगेज एन्झाईमची ऍक्टिव्हिटी होत असते त्यावेळी हे फॉस्फेट ग्रुप रिमूव्ह करायचं काम करत असतात हे बॅक्टेरियल अल्कलाइन फॉस्फेटेजेस. शिवाय एंड लेबलिंग करायचं असेल ज्याच्यामध्ये आपण देर आर टू मेथड्स, कॉमन मेथड्स ऑफ सिक्वेन्सिंग विच आर यूज. वन इज सॅन्जर्स मेथड, अनदर वन इज मॅक्झम गिल्बर्ट मेथड. तर मॅक्झम गिल्बर्ट मेथड मध्ये आपण एंड लेबलिंग करतो, 5' एंडला फॉस्फेट अटॅच करतो. तर तिथे सुद्धा बॅक्टेरियल अल्कलाइन फॉस्फेटेसचा यूज असतो आणि दुसऱ्या एंडलाच फक्त आपलं वेगवेगळे बेसिस ऍडअप होत जातात आणि आपण त्यांना आयडेंटिफाय करत असतो. So bacterial alkaline phosphatases, they show their activity at 65 degrees Celsius. They are sensitive to inorganic phosphates. So if inorganic phosphates are present in the medium, their activity will be reduced. Second one is calf intestinal alkaline phosphate, CIP. It is isolated from the intestine of calf and it catalyzes the removal of phosphate from the 5' end of the DNA or RNA. वरती बॅक्टेरियल अल्कलाइन फॉस्फेटेस काय करतो, हायड्रॉलिसिस फ्रॉम 3' 5' फॉस्फेट ऑफ डीएनए. हा calf alkaline phosphate, calf intestinal alkaline phosphatase removes phosphate from 5' end of DNA or RNA. It is very useful in gene cloning experiment to construct the DNA and to make the rDNA molecule that do not undergo self ligation. Self ligation केव्हा शक्य असेल, जेव्हा स्टिकी एंड्स तयार होतील. स्टिकी एंड्स नाही तयार झाले तर मग तसे स्टिकी एंड्स जनरेट केले जातात आणि त्यावेळी हे काफ इंटेस्टिनल अल्कलाइन फॉस्फेटेसचं मेजर रोल आहे. So treatment with calf intestinal alkaline phosphatase which do not have a phosphate groups at the 5' end of a vector, self ligation and circularization is not possible. तिथे हे phosphate group remove करायचं काम करत असतात. So this improves the efficiency of the vector and it can circularize properly. Next is shrimp alkaline phosphatase, SAP. Shrimp alkaline phosphatase is very specific heat labile, heat sensitive phosphatase enzyme, which is isolated from the Arctic shrimp Pandalus borealis. हा जो श्रिम्प आहे त्याच्यामधून हे आयसोलेट केले जातात. आर्क्टिक सर्कल्स मध्ये हे श्रिम्प सापडतात, पॅन्डालस बोरियालस आणि त्याच्यामधून हे एक्स्ट्रॅक्ट केलेलं हीट सेन्सिटिव्ह फॉस्फेटेज आहे, बट नॅचरल आहे. तो आर्क्टिक रिजन्स मधून आयसोलेट केलेला असेल, सो इट विल बी व्हेरी सेन्सिटिव्ह टू हीट. इट रिमूव्स phosphate from 5' end of DNA, RNA, DNTPs as well as proteins. SAP has similar specificity like calf intestinal alkaline phosphatase, but one difference is there that calf intestinal alkaline phosphatase can be irreversibly inactivated by heat treatment at 65 degrees for 15 minutes. पण सॅप्स हीट सेन्सिटिव्ह असल्यामुळे तो हीट ट्रीटमेंटने इनॅक्टिव्ह होऊन जाईल. But calf intestinal alkaline phosphatase, its activity optimum temperature 65 degrees असल्यामुळे no problem with temperature. That problem will be found with shrimp alkaline phosphatase because it is heat labile enzyme. So SAP is used for 5' dephosphorylation during cloning experiments, and it has various applications like dephosphorylation of 5' phosphate of DNA, RNA, for labeling the ends, end labeling साठी, self ligation of linearized plasmids. Plasmid जर सर्क्युलर फॉर्म मधून लिनियर फॉर्म मध्ये गेले असतील तर त्यांना परत सर्क्युलराइज करण्यासाठी सुद्धा SAP is useful. Shrimp alkaline phosphatase is useful. नंतर PCR product बनवण्यासाठी सुद्धा इट्स व्हेरी सिग्निफिकंट फॉर जनरेटिंग फॉर amplification of DNA. Then it can inactivate DNTPs from the PCR product. जे नको असलेले डिऑक्सिरायबोन्यूक्लिओसाइड ट्रायफॉस्फेट्स असतात, जे सुरुवातीला प्रायमर बरोबर अटॅच असतात, तर त्यांना काढून टाकण्यासाठी सुद्धा फॉस्फेटेज ला रिमूव्ह केलं की मग तेवढा पोर्शन निघून जातो. सो देअर ऑल्सो इट इज व्हेरी इम्पॉर्टंट. हिअर इट इज डाउनस्ट्रीम application. Then two primary uses of alkaline phosphatases in DNA modification is also there, restriction modification. To remove the 5' phosphate from vectors like plasmids, phages, which are treated with restriction enzyme. जे रिस्ट्रिक्शन एन्झाईमने ट्रीट केलेले असतात त्यांचा 5' फॉस्फेट काढून टाकायचं काम हे करू शकतात. दिस प्रिव्हेंट सेल्फ लायगेशन. कारण मग कसं होऊन जातं की वेक्टर कट केला आहे आणि तो परत सेल्फ लायगेट होऊन गेला तर तुम्ही त्याच्यामध्ये जीन ऑफ इंटरेस्ट हाऊ विल यू ऍड इट. So to prevent this self ligation, the phosphate should be removed from the plasmid, which is cut with the help of restriction enzyme. So it will not self ligate. You can then introduce the gene of interest there. So self ligation will be not possible because of the absence of 5' phosphate at the end. During ligation of the desired insert, the complementary ends of the insert and the vector will come to proximity of each other. आपोआप स्टिकी एंड्स असले तर रिस्ट्रिक्शन एन्झाईमने तुम्ही कट केलं असेल तर तिथे तो फॉस्फेट ग्रुप अवेलेबलच असेल डिझायर्ड जीन ऑफ इंटरेस्ट मध्ये. म्हणून इथून त्या वेक्टर मधून ते काढून टाकायचं, कारण मग तो राहील तर परत चिटकून जाईल त्याच्याच एंडला, ऑपोजिट एंडला. One strand of the insert has 5' phosphate will ligate with the 3' OH of the vector. 5' phosphate कोणाचा? जीन ऑफ इंटरेस्टचा. तिथे तो 5' फॉस्फेट एंड अवेलेबल असेल. इथून तो काढून टाकायचा वेक्टर मधून, म्हणजे ऑटोमॅटिकली जो 3' OH एंड आहे त्याला हा 5' फॉस्फेट जीन ऑफ इंटरेस्टचा लायगेट होऊन जाईल आणि नंतर तो अटॅच होऊ शकेल. आणि निक जो राहील, जो गॅप राहून जाईल फॉस्फोडायस्टर बॉन्डचा, तो लायगेज एन्झाईमने आपण नंतर भरू शकतो. लायगेज रिक्वायर्स ATP फॉर इट्स ऍक्टिव्हिटी, यू नो व्हेरी वेल. So it is used to remove 5' phosphate from the fragment of DNA prior to labeling with radioactive phosphate. That is again one important function of alkaline phosphatase. Very easy, simple and interesting. Next one is topoisomerases. Topo, because it is related with the topology of DNA, the name comes topoisomerases. So actually they are also known as DNA topoisomerases. They are used in overwinding or underwinding of DNA. Last semester we had seen DNA topology. So same तिथे सुद्धा topoisomerases आपण रेफरन्स घेतलेलंच होतं. Same enzyme is topoisomerase which removes the ओव्हरवाइंडिंग ऑर अंडरवाइंडिंग. पीळ राईट डायरेक्शनने किंवा लेफ्ट डायरेक्शनने ओव्हरवाइंडिंग झालेलं असेल तरी सुद्धा ते रिमूव्ह होऊ शकतो. देन वाय वी शुड रिमूव्ह दिस वाइंड्स? बिकॉज दिस वाइंडिंग प्रॉब्लेम may cause torsion or stress in the double helical structure of DNA, and that may damage the DNA. Usually at the time of replication, this is very common that when the replication fork opens the replication fork opens out, it creates stress or torsion at the other end of the DNA due to overwinding. आणि मग replication fork जशी जशी वाढत जाते तसं तसं stress create होत जातो. So if it is not removed, if that torsion or stress is not removed, it will stop the ability of the DNA to undergo polymerization आणि DNA replication ची process थांबू शकते किंवा स्लो होऊन जाऊ शकते. सो टू प्रिव्हेंट अँड करेक्ट these type of topological problems which are caused by double helical structure of DNA, topoisomerases are very important. असे प्रॉब्लेम्स कशाशी रिलेटेड असतात? डीएनए टोपोलॉजीशी, म्हणून त्याचं नाव दिलेलं आहे topoisomerases. So what these topoisomerases do, wherever there is torsion or stress created, they will bind to the DNA at that position and cut the sugar phosphate backbone एका स्ट्रँडमध्ये किंवा दोघं स्ट्रँड्समध्ये दोन्ही पॉसिबिलिटीज आहेत. एका स्ट्रँडमध्ये जरी कट केलं तरी पण तेवढा पोर्शन सुटून जातो. This intermediate break allows the DNA to be untangled or unwound. If it is overwound, it will be those winding will be loosened and unwinding will take place at this position. And once again, when the stress is relaxed, the DNA will be sealed with the help of ligase enzyme. Since the overall chemical composition and connectivity of DNA do not change, the DNA substrate and product are chemical isomers, differing only in their global topology. DNA substrate म्हणजे जो स्ट्रेस आहे, ज्याच्यात टॉर्शन आहे, तो ज्याच्यामध्ये ओव्हर बाइंडिंग झालेला आहे तो डीएनए सबस्ट्रेट असेल. आणि टोपोआयसोमरेसची ऍक्शन झाल्यानंतर टोपोआयसोमरेस त्या सबस्ट्रेटला टॉर्शन असलेल्या डीएनएला अटॅच होईल. मीन्स इट इज द सबस्ट्रेट फॉर डीएनए टोपोआयसोमरेस. असा सबस्ट्रेट रिलॅक्स होऊन जातो जेव्हा एक कट टोपोआयसोमरेस त्याच्यात करतो. सो दिस रिलॅक्सिंग विल रिझल्ट इन अ प्रॉडक्ट विच हॅज less winding than the substrate. So once again, you can focus here that the substrate and the product, that is overwound DNA and underwound DNA, both are just the topological isomers. They are nothing but the topological isomers. And they are only going to change the topology. They are not going to change any uh, uh, part of the DNA chemically. जो आधी होता तसाच डीएनए नंतर पण जाईल, फक्त त्यांचं वाइंडिंग जे आहे ते कमी झालेलं असेल. आणि म्हणून ह्या एन्झाईम्सला आपण टोपो आयसोमरेजेस असं नाव दिलेलं आहे. म्हणून सबस्ट्रेट अँड प्रोडक्ट हे दोघं आयसोमरच आहेत, फक्त एकामध्ये खूप वाइंडिंग आहे, एकामध्ये कमी वाइंडिंग आहे. Bacterial topoisomerases and human topoisomerases, they also show similar type of mechanism. They might manage the supercoiling of DNA. Topoisomerases have been further classified into subfamilies. In type 1 family, there are two subfamilies again, 1A and 1B, where enzyme links to 5' phosphate of DNA strand and 3' phosphate on the DNA. Type 1A topoisomerases मध्ये हा एन्झाईम 5' एंडला अटॅच होतो, फॉस्फेटला, 5' फॉस्फेट डीएनए स्ट्रँडच्या. आणि टाईप 1B मध्ये तो 3' फॉस्फेटला अटॅच असतो. एवढाच फरक आहे. टाईप 2 फॅमिली, द स्ट्रक्चर अँड ऑर्गॅनिझम डिटरमाइन्स द सबफॅमिलीज अँड द functions. प्रत्येकाचं फंक्शन कशावर डिपेंडेंट असेल, अकॉर्डिंगली दे हॅव बीन क्लासिफाईड. There are two types or families of this enzyme, type 1 family and type 2 family. Type 1 family passes one strand of DNA through a break in the opposite strand. In other words, DNA topoisomerase 1 enzyme cleaves only one strand of DNA. Attach तो एका स्ट्रँडला होतो, पण ब्रेक जो आहे तो ऑपोजिट स्ट्रँडला ट्रान्सफर करत असतो. सो इन दॅट वे वी कॅन से that type 1 topoisomerases are able to cut only or are able to make only single stranded cuts in the DNA. Type 2 family passes a region of duplex from the same molecule or a different molecule through a double stranded gap. दोन डबल स्टँडर्डच्या मध्ये जी जागा असते तिथे ऍक्च्युअली हे टोपोआयसोमरेजेस त्यांची ऍक्शन दाखवतात. सो दॅट इज वाय वी कॅन से in a common language that type 2 topoisomerases cleave both the strands of the DNA and result in double stranded breaks. That is the difference. Even if one strand is broken, unwinding will take place. Even if two strands are broken, more unwinding or fast unwinding will take place. That is the only difference. Topoisomerases can either relieve negative supercoils, both positive and negative supercoils, or induce positive and negative supercoiling of DNA. एक तर ते उलटी पीळ असेल, अँटी क्लॉकवाईज डायरेक्शन ती रिमूव्ह करतात, किंवा उलटी आणि सुलटी दोघं रिमूव्ह करतात, किंवा पॉझिटिव्ह आणि नेगेटिव्ह दोघं कॉइलिंग सुद्धा करू शकतात. फक्त कॉइल्स रिमूव्हच करत नाही, तर जिथे कॉइलची आवश्यकता असते तिथे सुद्धा हे एन्झाईम्स युजफुल आहे. The enzymes can also promote catenation. So what is catenation? When two single circular DNA strands are linked together after replication, that is known as catenation. And decatenation, the separation of two linked close circular chromosomes. म्हणजे डीएनए जे आहे ते रेप्लिकेशन झाल्यानंतर एक दुसऱ्याबरोबर वाइंड अप होऊन जातात. तर त्या कॅटिनेशनला प्रमोट करायचं काम सुद्धा टोपोआयसोमरेसेस दे कॅन डू इट. अँड if the two strands are linked to each other in a circular form or covalently closed circular form, then in order to remove that covalently closed circular form or open that covalently closed circular form or open the circular form, even then these enzymes can play their role. That is, जे entangling आहे त्याला रिलीव्ह करायचं काम हे करत असतात. So we'll see the summary of these topoisomerases and their role with the help of this diagram. Let me enlarge it. So type 1A topoisomerase आहे. हे टाइप 1B ची ऍक्शन होते. लिंकिंग नंबर त्याचा चेंज होतोय. इथे +1 होता तो -1 आहे, तो +1 झालेला आहे. इथे कट केलं, रिलीव्ह केलं म्हणजे ट्विस्ट ओपन झालेला आहे. सो ट्विस्ट ओपन झाला म्हणजे डीएनए वरचा स्ट्रेन कमी झालेला आहे. इथे परत सेम वे क्लीव केलं गेलेलं आहे आणि रिलॅक्स केलं गेलेलं आहे. सो दिस इज द रिलॅक्स. अल्टिमेटली बाय ब्रेकिंग दोज twists or removing those tangling or unwinding the DNA, you get a relaxed DNA. That is the role of topoisomerases. So here we finish with all the enzymes. Turning to cloning vectors. इतकं समजलं का? Before switching on to vectors, it is just revision. Even if you read it, you'll come to know. पण तरी पण एक वेळा फास्ट रिव्हिजन पण आपल्याला करणं आवश्यक आहे. इतकं समजलं? Should I start vectors? झोपले का? Jagruti, understood. Should I start vectors? Okay. So, different types of cloning vectors are used in rDNA technology. Plasmid vectors, lambda vectors, cosmid vectors, phagmid vectors, BAC, YAC, etc. So what is a vector? Vector is nothing but a small piece of DNA which is stably maintained in an organism and where we can insert our gene of interest or foreign DNA for the purpose of cloning or genetic engineering. What are the properties of vector? The vector may be taken from a virus or a bacteria or some other higher organism, not necessary always it should be bacteria and virus. The vector should contain features which will allow the insertion of फॉरेन डीएनए. ते कॅरेक्टरिस्टिक्स त्याच्यामध्ये असले पाहिजे, म्हणजेच रिस्ट्रिक्शन साइट्स असल्या पाहिजे, जेणेकरून वी कॅन क्लीव ॲट द रिस्ट्रिक्शन साइट, कट द डीएनए वेक्टर DNA and insert our gene of interest. Then it should be able to generate, usually sticky ends are expected, but not sticky ends. It can generate blunt ends, which can be converted into sticky ends. So that is also possible, so that we can use the DNA ligase to seal the two DNA fragments by enzyme ligase or formation of phosphodiester bond. Once the DNA fragment has been cloned in the vector, okay, so that recombinant molecule should be further transferred to another host cell. There are many cloning vectors, but the most common ones are the plasmids. And nowadays we know that we have artificial plasmids for that purpose. The first vector which were commonly used were some plasmids, bacteriophages, lambda, cosmid, M13, BAC, YAC, TI plasmid, etc., etc., etc. So this is the cos site of lambda. So here it can be cut. 12 बेसिस असतात 1 2 3 4 5 6 7 8 9 10 11 12. सो हे 12 जे ओव्हरहँग्स आहेत 1 2 3 4 5 6 7 8 9 10 11 12. 12 बेसिसचा ओव्हरहँग जनरेट करतो हा कॉस साइट ऑफ लॅम्बडा. व्हेरी व्हेरी सिग्निफिकंट. आणि लॅम्बडा खूप चांगला वेक्टर आहे, कारण तो इकोलायचा फाज आहे. सो इट इन्फेक्ट्स इकोलाय सेल्स. म्हणून आपल्याला स्पेशल ट्रान्सफॉर्मेशनची प्रोसिजर लागत नाही, की तुम्हाला तो वेक्टर विच कॅरीज द जीन ऑफ इंटरेस्ट त्याला परत लोड करा तुम्ही होस्ट सेलमध्ये बाय ट्रान्सफॉर्मेशन मेथड. ती गरजच नसते. All the cloning vectors, it is very compulsory that they should follow these properties, should have multiple cloning sites or polylinkers. जर क्लोनिंग साइट्स नसतील तर मग पॉलिलिंकर्स अटॅच होण्याची सोय पाहिजे, जेणेकरून आपण तिथे रिस्ट्रिक्शन एन्झाईमने कट करू शकू. क्लोनिंग वेक्टर्स may use topoisomerases instead of ligases. They can Another method of cloning without the use of DNA digest and ligase is by DNA recombination. The natural method of transformation or transduction or conjugation. त्याच्याने सुद्धा तुम्ही नवीन डीएनएचं कॉम्बिनेशन तयार करू शकता. तिथे डीएनए कट करायची गरजच नाहीये. So that is known as gateway cloning system, the natural cloning system. Once the gene is cloned into the vector, that vector should be safely not only introduced in the host cell, but that gene of interest should be properly expressed inside the vector or the host cell. If it is not able to express itself, means if it doesn't have the promoter for its expression, there is no use of that vector. Next, it should have a selectable marker. So every vector should contain a marker or a gene with the help of which we can select that particular vector from the other vectors. So antibiotic resistance is the common marker which is used, like beta lactamase gene, which confers resistance. It contains the beta ability to break down the beta lactam ring. Such beta lactam ring is present in which type of antibiotics? The beta lactam antibiotics, panem antibiotics ज्याला आपण म्हणतो. आणि त्यांची the beta lactam antibiotics are those which contain this beta lactam ring, like ampicillin, penicillin, cephalosporin, etc. The vectors there should be two selectable markers for antibiotic resistance. एक डिटेक्शन साठी आणि एक कट करून त्याच्यामध्ये जीन ऑफ इंटरेस्ट टाकण्यासाठी. Shuttle vectors have been decided. So these vectors can be able to be used in two different organisms. So for the purpose of cloning, we use one organism, and for the purpose of expression of that gene of interest, we use the other organism. So many vectors are selected on the basis that they have two selectable markers or two antibiotic resistance genes. So many of these are having resistance to zeocin, hygromycin B, or penicillin, tetracycline, etc. Oxotropic selection markers are also significant. So these markers will be some growth factor requiring markers like uh, biotin minus or biotin plus असे. तर असे मार्कर्स सुद्धा त्याला ऑक्झोट्रॉपिक मार्कर्स असे आपण म्हणतो. सो दीज मार्कर्स आर ऑल्सो सिग्निफिकंट अँड यू कॅन आयडेंटिफाय दी होस्ट ऑन द बेसिस ऑफ these markers. Kahi markers may involve the gene lethal to the host cells. So again, such markers are also used. Usually these genes are, those markers are such that they are normally causing killing effect on the host cells. Okay. But if you introduce your gene of interest in those markers, the killing effect will be converted into the non-lethal action, and that will be useful in detection. So लिथल जीन इंटॅक्ट सुद्धा राहू शकेल. जर तो इंटॅक्ट राहिला तर तो ऑर्गॅनिझमला मारून टाकेल. म्हणून मग आपण त्या लिथल जीन मध्येच आपला जीन ऑफ इंटरेस्ट टाकायचा, म्हणजे ऑर्गॅनिझम मरणार पण नाही आणि तुम्हाला त्याला डिटेक्ट पण करता येतील. सिलेक्शन ऑफ क्लोन्स विल ऑल्सो बी पॉसिबल. Then third thing is reporter genes are used in some cloning vectors. रिपोर्टर जीन्स नावावरूनच तुम्हाला कळेल की ते काय काम करत असतील. कुठेतरी ते स्क्रीनिंग साठी युजफुल असतील. सो दीज रिपोर्टर जीन्स आर युज्ड इन क्लोनिंग फॅक्टर टू फॅसिलिटेट द स्क्रीनिंग ऑफ successful clones, those which are transformed or which contain the rDNA molecule. Such type of clones need to be screened from the other clones, and for that purpose these reporter genes are used. Such features are present in cloning vectors, maybe LAGZ gene. So that will be useful. LAGZ gene will be a reporter gene. This LAGZ gene has the ability to produce enzyme beta galactosidase. So even in insulin, the LAGZ gene is used as a reporter gene. So when insulin is synthesized, there are various methods of synthesis of insulin. प्रत्येक जेनेटिक इंजिनिअरिंग कंपनीज आहेत जे रन करतात जेनेटिक इंजिनिअरिंग किंवा आरडीएनए टेक्नॉलॉजी. दे हॅव युज्ड different methods for production of this humulin. Humulin is the human insulin. Some have directly isolated the gene of alpha and beta genes. Others have introduced this gene of insulin next to LAGZ gene. So along with insulin, beta galactosidase is also synthesized. You can cut down this beta galactosidase enzyme by proteolactic activity and you will get the insulin protein. So these type of markers are very important. किंवा फ्लोरोसेंट मार्कर्स जे आहेत ते पण रिपोर्टर जीन्स म्हणून ऍक्ट होऊ शकतात. लुसिफेरेस जीन आहे तो पण रिपोर्टर जीन म्हणून ऍक्ट होऊ शकतो. Expression elements are very significant because even if you are successful in construction of a recombinant DNA molecule, transforming into a suitable host cell, in that cell it should be expressed with the help of these promoters and ribosome binding sites. So these are the expression elements for any type of recombinant molecule. That is a promoter which will express the gene and a ribosome binding site. Some may contain and they work as expression vectors. The target DNA may be inserted into the site that is under control of a particular promoter. So तुम्हाला तो सुटेबल प्रमोटर जीन असलेलाच वेक्टर तुम्ही सिलेक्ट करायचा ज्याच्यामध्ये तुम्हाला तो जीन ऑफ इंटरेस्ट टाकायचा आहे. म्हणजे मग त्या जीन ऑफ इंटरेस्टच्या एक्सप्रेशन मध्ये प्रॉब्लेम येणार नाही. Presence of promoter is very, very important. Without promoter, there is no possibility of the expression of that gene and you won't be able to detect it. One selection is blue-white selection. Already we have seen in FYBSc in the isolation of lac mutants where we have made use of this X-gal. So X-gal is a colorless compound and it can be broken down by beta galactosidase. So when it breaks down by beta galactosidase, it produces a dye known as dye indigo, which is blue in color. Xcal is structurally similar to lactose, but it is not the inducer of lactose operon. So we have to add an artificial inducer, IPTG, isopropyl thiogalactoside. So in that case, तुम्ही प्लेट्स अशा तयार करायच्या ज्याच्यामध्ये IPTG असेल, Xgal असेल, त्याच्यामध्ये तुम्ही इनॉक्युलेशन करायचं आहे. Xgal ब्रेकडाऊन जर झाला, कोण ब्रेकडाऊन करू शकेल ज्याच्यामध्ये बीटा गॅलेक्टोसाइडेज जीन इंटॅक्ट असेल, ते ब्रेकडाऊन करू शकतील. ज्याच्यामध्ये बीटा गॅलेक्टोसाइडेज जीन मध्ये जीन ऑफ इंटरेस्ट इन्सर्ट झालेला असेल ते करू शकणार नाही एक्सगॅलला ब्रेकडाऊन, कारण बीटा गॅलेक्टोसाइडेज तयार करणार नाही. इन दॅट केस some colonies will be blue, some will be white, so you can select those colonies which are of your interest. Cloning vectors without promoters or ribosome binding sites for cloned DNA segments are sometimes used. For example, cloning genes produce some toxic substances. There also we can make use of such type of promoters. Some vectors are designed for transcription only with no heterologous proteins expressed. So here transcription of only the gene of interest will not take place. Similar proteins gene of interest सिमिलर प्रोटीन तिथे तयार होऊ शकणार नाही जरी त्याचा जीन प्रेझेंट असेल. These vectors, they are known as transcription vectors. So they do not contain the sequences necessary for polyadenylation, termination, and they may not be used for protein production. Clear? First, we'll see the plasmid vectors. Plasmids are vectors because they can be modified and replicated. They can be artificially engineered in a laboratory. They have origin of replication. They have self-replication capacity. They have the sites, multiple cloning sites or restriction enzyme cutting sites. Several cloning sites are present. They have selectable markers. So you can screen them or detect them easily. They may, so you can introduce the promoter regions in the artificially or genetically engineered vectors, and म्हणून plasmids are very good vectors. So you can cut them with at specific points with the help of restriction enzymes, which are molecular scissors, and you can generate the sticky ends. Then you can insert the gene of interest with the help of DNA ligase and construct a recombinant molecule, introduce that recombinant molecule into the host by transformation. And if this plasmid contains the promoter, it will be expressed in the host cell with the help of this promoter. The first plasmid which was used as a cloning vector was PSC101 of Salmonella typhimurium. It is known as the first generation plasmid. MCQ मध्ये हे विचारलं जातं, नोट डाऊन करून ठेवा. फर्स्ट जनरेशन plasmid vector PSC101. सो याच्यामध्ये त्यांनी फ्रॉगचं जीन त्याच्यामध्ये इंट्रोड्यूस केलं होतं PSC मध्ये, आणि साल्मोनेला टायफिम्युरियम मधून त्याला आयसोलेट केलेला होता तो वेक्टर, आणि त्याच्यानंतर मग इकोलायचे प्लाझमिड्स क्लोनिंग वेक्टर म्हणून वापरले गेले. So first generation plasmid vector is PSC101 isolated from Salmonella typhimurium. Next one is PBR322 plasmid, B for Bolivar, R for Rodriguez. It's a second generation plasmid vector. The main characters, it's a constructed plasmid actually. PBR322 was constructed in the in the laboratory research lab of America by Bolivar and Rodriguez, and hence the name is given to it. It consists of multiple restriction sites for Bacillus amyloliquefaction restriction enzymes, Haemophilus influenzae restriction enzyme, Streptococcus albus restriction enzyme, Proteus vulgaris restriction enzyme, Pseudomonas Providentia Stuarti restriction enzyme, and equarmin and other restriction enzymes. It contains selectable markers, ampicillin resistance and tetracycline resistance gene. It has origin of replication, and it has the replication ROP gene, which is very necessary for the processing of replication. Different antibiotic resistant genes, they act as restriction sites where you can insert the gene of interest and ligate the gene of interest, then transform that vector into the host cell and express the gene in the host cells. Alternative selectable markers are also present, so they can produce some color, that is a chromogenic substrate if you are using. Initially it is colorless, then you can add it. XGAL is the best example. Usually it codes for the LAGZ gene, it codes for enzyme beta galactosidase. So when foreign gene is inserted in the gene for beta galactosidase, then beta galactosidase gene will be inactivated. आणि क्रोमोजेनिक सबस्ट्रेट तुम्ही जेव्हा एक्स गॅल वापरतात तेव्हा तो त्याला ब्रेकडाऊन करू शकत नाही, ब्ल्यू कलर कॉलनीज तयार करू शकत नाही, कलरलेस कॉलनीज मिळतात. Colorless colonies is an indication that your gene of interest is present in the vector. So this is the diagram of PBr322 plasmid. 4.36 kilobases of plasmid. It has the restriction site PST, EcoR1, HIN3, BAMH1, and this restriction enzyme, ampicillin resistance, tetracycline resistance, origin of replication. Okay, so several restriction sites are also present. The next generation plasmid, it is the PUC plasmid, PUC 18. University of California मध्ये त्याला isolate केलेलं होतं. It is known as third generation plasmid vector. हे फार इम्पॉर्टंट आहे बरं, तुम्हाला माहिती पाहिजे फर्स्ट जनरेशन मधला कुठला एक्झाम्पल आहे, सेकंड जनरेशन मध्ये कुठला आणि थर्ड जनरेशन मध्ये कुठला. PUC plasmid is a modified PBr322 plasmid. तो 3.6 किलो बेसिसचा होता. त्याला नको असलेला पार्ट, it was just cut down just to remove the bulkiness of the PBr322 plasmid. It was cut down, the unwanted portion was removed, and it was reduced to 2.7 kilo bases. It has a high copy number than PBr322. It can produce 500 because the size is reduced. It can produce more copies per cell. P stands for plasmid, UC for University of California, and was first developed by J. Messing et al. Several series of PUC plasmid are known: PUC8, PUC18, PUC19. The whole even origin of mutation of PBr322 is modified in E. coli, and that is why its copy number is increased. It has ampicillin resistance gene, just like PBr322. It has LAGZ gene, that is the multiple cloning site. You can use the insertional inactivation of LAGZ gene. So the cell loses the ability to produce beta galactosidase. You can use blue-white screening for detection. They can be used in cloning vectors as well as expression vectors. What are the advantages of PUC over PBr322? It has high copy number. It has one-step selection possibility. And these restriction sites, they are clustered or they are present close to the multiple cloning sites. Because the unwanted portion is removed, these sites have come close to each other. Disadvantage is its size it cannot be used for those genes whose size is greater than 15 kilobases. Because already it is 2.6 kilobases, how much it is going to hold? 15 किलोबेसिस पेक्षा मोठा जर जीन ऑफ इंटरेस्ट असेल तर त्याला तिच्यात टाकता येणार नाही, कारण मग प्लाझ्मिड छोटा आणि जीन ऑफ इंटरेस्ट मोठा असं होऊन जाईल. दॅट इज वाय इट कॅन बी यूज फॉर लिमिटेड साइजेस ओनली. विल स्टॉप हियर, कंटिन्यू द लेक्चर मोस्ट प्रोबॅबली एक दिवसाआड घेऊ, कारण MSc 1चं प्रॅक्टिकल सुरू आहे, TYBSc चं लेक्चर सुरू आहे, SYBSc चं थिअरी सुरू आहे, सगळं ऍट अ टाइम सुरू आहे. सो प्रॅक्टिकल सुद्धा घ्यायचे आहेत. MSc 1चे प्रॅक्टिकल्स 5 होऊन गेलेले आहेत. अजून 3-4 झाले की मग तुमचे प्रॅक्टिकल्स पण मी सुरू करेन. ओके, सो विल स्टॉप हियर, कंटिन्यू विथ द नेक्स्ट वेक्टर्स टुमॉरो. टुमॉरो और डे आफ्टर टुमॉरो मी तुम्हाला इन्फॉर्म करेल. If you have any difficulty, you can ask me on the group also. Or you can even call me. No problem.